Hello and welcome everyone to this podcast on cell biology. In this podcast, and the next several podcasts, we're going to talk about cell membranes. The first couple podcasts will focus on lipids in the membrane, so lipid bilayer. And that'll take a couple podcasts to get through all that. And then we're going to move on to membrane proteins. But let's go ahead and get started with the lipid bilayer. So the lipid bilayer. So let's begin by drawing a very simple cell here. So let's list some of its functions over here. The first is a fairly straightforward function, and that is it defines boundaries. In a sense, it determines what's inside the cell and what's outside of the cell. In doing so, it acts as a permeable barrier. Because of the nature of the membrane, we'll talk a lot about the nature of the membrane. Some things can get through the membrane. Some things can get through going back outside of the membrane. Some things can't get out of the cell because of the membrane. And some things can't get into the cell through the membrane. So it serves as this permeable barrier defining what's in and what is out of the cell. 2, on the membrane itself, it's the site of specific proteins and specific functions. We'll learn that there are certain proteins unique to the plasma membrane. Maybe even one side of the plasma membrane versus the other side of the plasma membrane. We also know from previous classes that within the cell there are different structures. We might have the mitochondria, the nucleus, and all the other organelles that we've discussed. Within each of these organelles, there are specific proteins that are specific to the membranes that are associated with these organelles. And with that in mind, we can talk about specific functions. Each of these membranes has a specific function. The membranes in the mitochondria are different than the membranes on the plasma membrane, as the nucleus or the Golgi or any of the organelles. So they help define specific functions. They regulate transportation. There are specific proteins on the membrane that are unique to specific organelles that allow the transport of molecules from the outside of the cell to the inside, or from the inside out of the cell, or into specific organelles, maybe out of specific organelles. The properties of membranes sometimes allow transportation, as we mentioned up here, it being a semi-permeable barrier. It can allow movement without the use of a protein as well, for some solutes. Our fourth function, detects and transmits electrical and chemical signals. Various chemicals in this outside of the cell will bind to this outer plasma membrane and cause some sort of downstream signal to occur. Maybe it will be important in telling the nucleus to divide. Maybe it'll be important in telling the cell not to divide. Various kinds of signals from the outside of the cell to the inside of the cell are detected by the plasma membrane and other membranes. Okay, now the fifth function I want to talk about, and our last one, is that the membrane can serve the function of cell adhesion and communication. I can't draw the whole cell here, but we may have a cell here, like so, and these two membranes will connect with each other. They will adhere to each other. And there are very specific proteins that are involved in uniting these two membranes so that these two cells are next to each other. We'll talk about those kinds of junctions later, but some are called tight junctions, some are called occludins. In addition to that, some of these junctions known as gap junctions. We'll talk more about those later. But they allow for the transport of small chemicals from one cell to the other. So those were just some of the functions of the membranes. But now I want to get more into membrane, more appropriately, the lipid part of the membrane structure. As we begin this discussion, I want to talk briefly about the differences between eukaryotes, archaea, and bacteria. So let's begin by talking a little bit about bacteria. Remember that bacteria are either gram-positive or they're gram-negative. Remember that the positive bacteria, gram-positive bacteria, are characterized as having a very thick peptidoglycan layer. And for the gram-negative bacteria, it has a very thin peptidoglycan layer. So we could draw the peptidoglycan gram-positive bacteria like such, with a very thick peptidoglycan layer. And for the gram-negative bacteria, we can draw it as a very thin peptidoglycan layer. The gram-positive bacteria will have this inner membrane, much like the plasma membrane that we'll think about when we talk about animal cells. The gram-negative bacteria with this thinner peptidoglycan layer will have an outer membrane and an inner membrane. All of these membranes, shown here in red, will have similar characteristics that all membranes share. One thing different about the gram-negative bacteria is they will have this lipopolysaccharide layer here. So it's just like a membrane, but it has a very high content of these lipopolysaccharides. So what does this peptidoglycan layer do for the gram-positive bacteria? Well, it does make them more resistant to drying out and to physical disruption to the membrane. Whereas this thinner peptidoglycan, surrounded by these two membranes here, are more susceptible to physical damage and are more likely to dry out. However, the gram-negative bacteria are more resistant to antibiotics. So let's write that here. More resistant to antibiotics. I'm just going to put AB for antibiotic. One of the reasons that they are more resistant to antibiotics is because of this outer membrane that contains these lipopolysaccharides. This outer membrane protects against certain chemicals and antibiotics. And as we go through this in the next podcast, we'll talk a lot about the structure of these lipid membranes. But the main take-home point from these bacteria that I want you to take is that gram-positive bacteria contain a single membrane, whereas gram-negative bacteria contain two. And this second outer membrane is what helps protect the gram-negative bacteria against certain antibiotics, and ultimately makes these more likely to be antibiotic resistance from a clinical aspect. Now, next I want to talk about something that happened here in the evolution of archaea that makes their membranes quite a bit different than bacteria and eukarya. So if we think about this evolutionary context, the common ancestors that gave rise to the bacteria and all of these species here that are no longer with us, and ultimately to our eukaryotes, are similar. But something happened here during this branch that gave rise to archaea that produced a slightly different but significant membrane structure. So what is this difference? Well, to explain this difference, I want to go back to something I said at the beginning of the semester when I talked about lipids, but more specifically, phospholipids. And if you remember, we said that they contain this glycerol molecule. And on this glycerol molecule, there was a phosphate group attached to one of the carbons. And then to the middle carbon, there was a hydrocarbon chain. And to this top carbon, there was another hydrocarbon chain. But they were connected to these hydrocarbon chains by an ester linkage. And I didn't draw the ester linkage at the beginning of the semester. At least I don't think I did, but let me draw it now. And then here's our chain of hydrocarbons. And remember, at each of these carbons, where the points are, I won't draw them all, remember there are hydrogens attached. Either one or two, depending on if it's saturated or unsaturated. We'll come back to that later. But for now, I'm just going to put 2 hydrogens there. And this one would look similar. We'll learn that they might be different lengths here. They'll have a different degree of saturation, but that all comes later. It's still carbons with the hydrogens attached. So I mentioned that an ester bond was formed here, but so let's remind ourselves what an ester is. It's a carbon with a double bond O, an oxygen attached to an R group, and then an R group over here, a different R group. And that's what we see here. We have a carbon, double bond O, an R group here, an oxygen here, and an R group over here. So, again, this is what we see with bacteria and eukaryotes. So, what do we see with archaea? Well, you're going to see that it's somewhat similar, but the difference has quite a significant impact. We still start off with our glycerol molecule here, with the phosphate group down here, but instead of forming an ester bond here, what we're going to form is an ether bond. And essentially, if I can remind you here, an ether is an oxygen with an R group on either side of it. So what we're going to have here is an oxygen. Okay, so this here, let me just write ether. That's the first difference. There's one other difference with archaea, and that is how the chain forms. Here we have repeating units of CH2, 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 and down the line. Here what we have, CH2 here, comes down here to a CH, and then it has a CH3 here. CH2 here, CH2, CH2, and then we have another CH3. And we can keep repeating this all the way down for as long as we want to go. And same thing on this strand here. So you can see this adds quite a bit of a different structure to these hydrocarbons that extend down these lipids. Why does it have these differences? What is it about these differences that allowed archaea to develop? We know that archaea are often found to be extremophiles. So did this allow for the membrane structures to evolve in such a way that allowed them to withstand high temperatures, other extreme conditions? I don't know. Maybe we'll find that out later on. But for now, what I want you to know are a few things that we talked about. We've already talked about the bacteria, how they're different with the outer and the inner membranes. But now I want you to know that in archaea, there are 2 key differences in their lipids. The first is that they form this ether bond here, as opposed to the ester bond over here in the bacteria and eukaryotes. I also want you to know that the repeating hydrocarbon structure of these tails is different, mainly in that they form an extra carbon link in repeating intervals along the tail. And finally, I want this to be just another reason, another example for you to consider why we no longer classify bacteria and archaea in the same group. We'll point those out every now and then throughout the semester, but it's important that we continuously recognize that these are 2 different phylogenetic organisms. Now, on the previous slide, I drew in pretty good detail what a lipid molecule would look like. But I didn't point out some key features that I think are relevant here. And so I'm just going to draw a generic molecule right now and point out some features that all of these lipid molecules will have. They all have a hydrophilic portion, or we might call it a polar portion. And the other part that all lipid molecules have is the hydrophobic portion, or we might call it the nonpolar portion. Remember that water will interact with this hydrophilic portion. And I also want to remind you, any molecule that has both a hydrophilic and a hydrophobic portion, we call those molecules amphipathic. Now, on the last whiteboard, I showed you a phospholipid, and here I just drew a generic lipid, but the lipids can be quite different. And I'm going to draw 3 classes of lipids. The first one is going to be phospholipids. I'll start with that one because we have some experience with this one from the last whiteboard and previous in the semester. And the phospholipids will have that glycerol unit like so. it will have this phosphate group here. I drew it up here so we can see that's where our hydrophilic portion is, right there. So this could be a phosphate group. And then it's going to have these hydrocarbon chains. And I drew this as we would see it in eukarya or bacteria, not archaea. Now something I want to point out to you is that these generic phospholipids can be modified just to thicken the plot a little. So attached to these phosphate groups, we could add a serine, or we could add a choline. Choline's a good one to think about because it's one of the most common found in membranes, choline. So they can be modified to some extent. The next type of lipid molecule I want to talk about are sterols. They will have this basic 4-ring structure as I've drawn here. You'll notice a few features. One is the sterols all have these 4 rings, and they will all have this hydroxyl group up here. I want to make sure I point that out because this is the hydrophilic portion of the sterol right there. So remember, all lipids are going to have a hydrophilic and a hydrophobic portion. Hydrophilic, hydrophobic. You'll also notice that these rings, there's 3 6-membered rings here, and then a 5-membered ring here. You're never going to have to draw this, but it would be good if you could recognize it. Now, just like this phospholipid could be converted to various types of phospholipids by adding serines or cholines, and the sterols are the same way. They can also be modified to contain certain structures here. There may be some form of hydrocarbon tail added here. And I'm not going to draw the specific nature of it, but one of these that can be added would make this sterol cholesterol. Remember, cholesterol is only found in animal membranes, and we'll come back to that in a little bit as to why this is important in membranes for animals. Okay, the last class I want to talk about are glycolipids. Glycolipids are very similar to phospholipids. They will have our carbon glycerol backbone here. To 2 of these will have a hydrocarbon chain, like so. But attached to this top one are various structures. And these structures, what characterizes them is that the bond that holds them together is a glycosidic bond. Just like we see when we take sugars and build larger polysaccharides. The bond that holds those together are glycosidic bonds. And so often what's added here are sugars. Galactose could be added here. So what is their purpose? Why do we have these? And these are primarily found in eukaryotes. And what we have on a eukaryotic cell here are these glycolipids. And these glycolipids will have various sugars attached to them. It could be strings of sugars. And they serve several purposes. They're important in our immune system, identifying cells that are our own cells versus cells that are not our cells. They're important in our blood types. What makes us type A, type B, type O, all depends upon what kind of sugar moieties we have on the outside of these glycolipids. And they're also important in cell-to-cell interactions. Glycolipids are also found in bacteria. So in bacteria, they're found also on the outside of the cell, and they help form large interactions here with other molecules. And we know they're important because if we mutate the synthesis of these glycolipids in bacteria, they actually do turn out to, for the most part, be viable. There might be some exceptions to that. They have an altered shape, that is, they're misshaped. They have a motility defect. They're not as motile. And lastly, they seem to have an increased antibiotic sensitivity, more sensitive to antibiotics. So they appear to be quite important as we look at both eukaryotes and bacteria. Now I want to take some time and draw the lipid bilayer, the whole lipid bilayer. These lipids, phospholipids or glycolipids, will all have their polar heads on the outside with their nonpolar tails on the inside. So we might have the cytoplasm here and the external environment here. In either case, there will be water molecules on both sides here interacting with these polar heads, the hydrophilic portions. We might as well just go ahead and label these while we're doing this, keep everything legal. These are our hydrophobic portions, and then these are our hydrophilic portions. And as I said, in an aqueous solution, we have our water here and here, which forces this interaction. The hydrophobic come together because they're not interacting with the water. The water sort of, in some ways, pushes these hydrophobic parts of the membrane together and stabilizing the hydrophilic portions out here with hydrogen bonds with the water molecules. Now I want to draw this in a slightly different way here. What if this lipid was just a straight sheet? It still had these components here, but they're wrapped up in this thin line. Because we want to continuously keep water interacting with these polar heads, and the hydrophobic portions are avoiding the water, so water isn't going to come along here and interact with these hydrophobic portions, the membrane has to bend to kind of exclude that water. And so what happens is, if I could draw down here, we end up forming this kind of arch structure. And it's going to continue to grow because continuously, as this membrane moves around, growing, avoiding the water, so to speak, eventually what's going to happen is they're going to form this circle, or what you and I happily call a cell. The need for the hydrophobic components to interact with each other and exclude the water forces the membrane to kind of bend around, pushing together until you form a locked structure where we still have water on the inside and outside, but not interacting with the middle here. Your book uses this phrase that I like quite a bit. It's called prohibition on free edges. And essentially, we can think of this in a few ways. One way is just as we described. It doesn't like these free edges. We would call these free edges. The cell doesn't like that. And it doesn't like it because it's energetically unfavorable. It's unfavorable because we know that water interacting with hydrophobic molecules is an unfavorable interaction. So it's going to do what it needs to do to make it a more favorable interaction and prohibit these free edges from forming until eventually you form a cell that we see here. The same could happen when this cell becomes damaged. So let's just damage the cell here with my finger. And right now we have two free edges. This is energetically unfavorable for the cell. And so what the cell will do is immediately make the interactions of this membrane with the aqueous solution around it more favorable by filling in these gaps with new membranes. Again, going back to this prohibition on free edges. Essentially, this is to make the interactions of the aqueous environment with the membranes more energetically favorable. I think this is a good place for us to stop for this first podcast on the lipid bilayer. In this podcast, we talked about the differences of lipids between archaea, bacteria, and eukaryotes. We also talked about different lipid membrane components and how those were important. And we discussed how ultimately that lipid is formed to prevent those free edges. In the next podcast, I'm going to talk more about the flexibility and in particular the fluidity of the membrane and how things move in the membrane and then how membranes are formed. All right, so that's it. If you have any questions, let me know. If not, I'll see you on the next podcast. Bye.